Okay, fantastic, guys. Welcome to Monday afternoon. And my name is, it is recorded, so I'm just going to say my name is Vera Silverman. And today I have an absolutely distinct privilege and honor to host again Dr. Kiran Krishnan. I think this is number 4 lecture. Kieran is an amazing dude, and I'm sort of becoming familiar because I asked him once, "How do you want me to introduce you?" He said, "Super nerd." So the interesting thing is, this super nerd is an amazing genius nerd. That's what we need. But this nerd has a heart. He absolutely, in his business and what he does for the world, honestly, guys, what he does for the world, he absolutely could have said no to me. So the thing is, I'm trying to learn health from every perspective I can. And a couple of days ago, I was on a webinar by an incredible Dr. Jeffrey Bland, who is the godfather of functional medicine. There were a whole bunch of distinguished people on board, and there was one thing that was told, that there are two most important things for our health. It's gut and microgut. And I entirely agree with this. So, what happened today, Dr. Krishnan is going to talk to us about evaluation of our microgut, because it's interesting. We know we started finally, we're doing individualized medicine. So we are examining our genome, our epigenome, our metabolome, our proteome, whole bunch of things that are related to our cellular health. However, our cellular health may be entirely dependent on our co-host and our co-family, which is our microbiome. So we are starting to evaluate our microbiome, not like we used before with the blood occult test and something that we were testing for oven parasites. And but we're doing finally the individualized and precise evaluation, and microbiome labs are on top of things. So I am your victim, and Dr. Krishnan is our professor, Dr. Extraordinary. Kiran, it's all yours. Thank you.
Thank you so much. Really appreciate this opportunity, and I'm super excited to be able to talk about biome effects. So, as Svetlana said, that the analysis of the microbiome can really provide some really important insights into your health and wellness, because the fact is it's your microbiome that controls much of your outcome, right? The microbiome houses the vast majority of the genes that your system uses. The microbiome houses the majority of the metabolic processes that we count on. The microbiome dictates how our body responds to food and the environment around us. So the microbiome is really in the driver's seat. And it isn't till most recently that we started recognizing that. And then now we have some ways of getting some insight as to what our microbiome looks like. And that's what Biome Effects is for. Now, Biome Effects has changed the game when it comes to microbiome analysis, because there's been other kits on the market over the last 10 years. However, they're using antiquated technology. There's a type of sequencing called 16S ribosomal RNA sequencing. That's a very low cost way of running sequencing analysis on the microbes that live in your stool sample. The problem there is that the technology is quite inaccurate in order to identify all of the species at the species level, right? So it does a pretty good job of identifying bacteria to the genus level. If people aren't familiar, genus is like, let's say you're familiar with Lactobacillus acidophilus. The Lactobacillus is a genus and Acidophilus is a species, right? So it could be any one of 40, 50 different kinds of lactobacilli. You won't know exactly which kind and what it means and what the functionality is until you can identify it at the species level. So these other tests on the market cannot look at the microbiome accurately at the species level. And that's the most important part of the analysis, because once you can identify all the bacteria present at the species level, then you can create a map of the ecosystem, because the mapping of the ecosystem is what really gives you what the outcomes will look like within your microbiome, because it's not just the singular absence or presence of individual species, it's who else is in that ecosystem and how those microorganisms work together or against one another. So there's a functional component to it that comes about only from understanding the bacteria at the species level and then mapping the microbiome based on that species level identification, right? It's the ecosystem that matters. It's all of the players taken into context that matters. It's not singular pathogens that matter. It's what else is there, who else is influencing the metabolic processes, the genomic processes, and so on within the microbiome. So Biome Effects was designed to be the first functional microbiome analysis because it provides species level identification, and we use the best in this field. Cosmos ID, which is a lab that we've partnered with, who's founded by Dr. Rita Caldwell, who is the absolute stellar researcher in this space of microbiome ecosystems and mapping. She has over 800 published papers in this space. And so we worked with them to really come up with the most functional analysis. So what I'll show you is the way the report lays out the information about your microbiome. It starts off with a high 30,000 foot view of your microbiome with some of the parameters that are critically important to overall health and wellness. And then it digs down deeper and deeper into aspects of the microbiome that are functionally off and functionally can be changed. That's the other good thing about this test, is that everything that's tested for, all the endpoints that are measured, can be actionable. They all have a lifestyle component, a dietary component, a dietary supplement component that can impact the outcomes. So now let's take first the report summary, the overall view. So there's a few things here that are being measured. Number one, this is the overall index of the microbiome. That's taking into consideration all of the scoring, all of the analysis, and it says where is your microbiome in a bell curve from really off to really healthy looking microbiome. This one is right in the middle, right? So it's the index is right about half. So it's almost like the 50th percentile. Now, when we dig into some of the other aspects, it'll say, this will give you a clue as to why it's not 90th percentile, 85th percentile, why it's right in the middle. One of the first things to note, and probably the most important feature of a healthy microbiome, is diversity, right? So the diversity score is relatively low here. We want the diversity score to be as high as possible. And again, we'll dig into the diversity more specifically on the next part. Beta diversity and alpha diversity are two different types of diversity. I'll talk about what they are, but beta diversity is relatively high. And most people in the US, you'll find beta diversity being high. And again, I'll explain in the next page what that means, but alpha diversity is really the key. Alpha diversity tells a lot about the health of the overall microbiome. And because alpha diversity is low, this one called the resistome is also low. The resistome is a measure of resistant forces within the microbiome. That is, how strong is the competition within the microbiome. This tells you how stable the microbiome is. So the higher this number, the more stable the microbiome is, the more resilient it is to things that may disrupt it, right? A course of antibiotics, or eating poorly for a few days, or travel overseas, things that may cause a disruption to the microbiome. The microbiome is more resistant against those changes if this number is higher. And then finally, the pathogen index, right? It's not at all abnormal. In fact, it's quite normal to have pathogens within the microbiome. And in fact, some pathogens play an important role in the functionality of the microbiome as long as they are under control, right? If their numbers are being controlled by the commensal bacteria, then the pathogens are not really driving any issues. One of the things I really dislike about the vast majority of microbiome tests on the market is they have such a huge focus on pathogens, and that's primarily what they look at. Then they also do things like PCR on the pathogens, which means they artificially amplify the amount of pathogen that's present. So it makes everything look high from a pathogen perspective, when in fact the presence of the pathogen may just be there and the pathogen may be relatively low compared to the rest of the microbiome. So, which means that the pathogen is not really an issue, which means you don't need antimicrobials and so on. What this index is telling us is we want the pathogen index to be somewhere here in the 6 to 10 level, better around 8. This means that pathogens may be an issue and it's something worth looking into, right? These are the keystone species, some of the most important groups of bacteria within the microbiome. All of these organisms are associated with health and wellness. The higher their levels, the more protected the gut is. All of these have to be worked on. These are all low, and the good news is most of these can be increased through diet, supplementation, and so on. And then these are functionalities within the microbiome, the things that the microbiome produces that are critically important, that are also low. And this is not surprising because many of these things that are critically important, like saccharolytic fermentation, butyrate production, GABA production, vitamin biosynthesis, and so on, all of these things are actually functions of these important keystone species. So as these keystone species are low, these functionalities are going to be low as well. So this gives a big 30,000-foot view on what's going on overall in the microbiome. So let's dig a little bit more into some of these specific things. So now the first thing we look at is alpha diversity. So alpha diversity tells you how many different species are within the microbiome sample, right? So here the test picked up 101 different species. Now to put that into comparison, if we were to test the alpha diversity of the current hunter-gatherer tribes, like if you look at the Hadza tribe in Tanzania, the tribes in Papua New Guinea, their absolute number of species would be somewhere close to 400 different species, right? So as a population, we've lost a lot of organisms that should be commensal within our gut over the decades of the lifestyles that we put ourselves in and we live, right? We live away from the environment. We don't engage in the natural environment as much. We're not getting exposed to as many bacteria as our ancestors or most recent ancestors do. And so we are losing many of these microbes. And then, of course, we surround ourselves with lots of antimicrobial exposure, whether it's through food, water, antibiotics, and so on. So it harms our alpha diversity. Now, this plot is actually, like, you can look at this as a bell curve. So this is the 0 percentile. This is the 100 percentile. This is the peak of the bell curve. So average North Americans, their diversity is somewhere here in the middle, about 125 species, right? So this sample is a little bit below average. We want to make sure we're getting this number up. That's really critical. But even the average North American, the diversity is really low. Even if you were at the high end, the 99th percentile of this curve, we're just at about 200, which is good. But remember, our microbiome should be in closer to 300 species. So diversity is something we always need to work towards. And virtually every study on this subject has shown that diversity in the microbiome is critically important to overall health and wellness. Beta diversity tells you what your collection of organisms looks like in comparison to the population around you, right? So beta diversity being high means that the collection of organisms fits well within the types of organisms found in healthy normals around that same geography. If the plot was all the way here in the corner, it would say that your beta diversity is way off, which means that there are organisms within your microbiome that are not well suited to that ecosystem that you're in right now. Maybe you went and spent a year in the Amazon jungle and you picked up a bunch of organisms that are completely different than what's here, and you're suffering from issues that may be because of low beta diversity, which means that you've got organisms that are not well suited to the new environment that you're in, right? So most people that we test, the beta diversity is relatively high. That means their collection of organisms fits well within their ecosystem, and most people have relatively low alpha diversity. So this is something that almost everybody we test needs to work on. Because the alpha diversity is low, then the resistome is going to be low as well. We want to be here at 1. This is around 0.3. And so we want to bring this up as well. And this will come up as you improve the diversity here, because it's improving resistance forces and making the microbiome more resilient in general, right? So that's how that works. Now, this is the same thing, a visualization of some of that other data. What's good here is the proportions of the different groups of bacteria that are found in the sample are very similar to the proportions in the healthy normal population, right? So for example, if you look at, this is the US healthy normal population, 63% of people's microbiomes belong to the phylum Bacteroidetes. In this case, 63% on the dot belong to phylum Bacteroidetes, 27% to Firmicutes, here pretty close, 29%. Same thing, 2.8% to Proteobacteria, in this case a little bit higher, but it's not a big issue. Actinobacter is at 2.6%, here it's 4%. So it's pretty close in terms of the types of organisms. This sample just needs more organisms within each of these groups, right? So
Can I
that is
interrupt
part.
you for a second, please?
Uh-huh.
Because you're talking about diversity, absolutely, clearly and understood. Now, let's say the question is, taking certain probiotics for a very long period of time, basically similar probiotics, will that affect the diversity? So for example, let's say I take lacto, different species of lacto and different species of bifidum. Thus the sample shows lesser diversity with very small amount of lactose and bifidum. Can it be attributed to the fact that, I've got tourist in my bowel?
Yeah. So
hang on. And same thing with the Saccharomyces. So I'm consistently, for example, taking Saccharomyces, let's say, so, okay, sorry, going personal because whatever, that's what we're doing, I'm sacrificing, I'm a sacrificial lamb. So, no, I'm sacrificial poop. Anyway, so hope everybody's laughing. So anyway, so taking spore biotics, taking probiotics with bifidum and lacto for multiple reasons, especially for the production of short chain fatty acids and vitamins and GABA, basically, whatever you described as kind of low, and then taking Saccharomyces for the cleanup thing, but consistently for a long time. Will that affect diversity and can it be rotated? Because I also heard from Dr. Bergson that probiotics are the very expensive visitors. They're tourists for a short period of time and then they're leaving.
Yeah. So the vast majority of bifido and lactose species and bifido and lacto probiotics you take don't survive in the gut, right? And if they do, they will compete with your natural bifido and lacto. So they can actually affect diversity in a negative way over time, right? Some of them can be very useful for particular effects like Rhamnosus GG or GR1 or Acidophilus DDS1. They have very specific functionality because of their very unique subspecies of those organisms. But those general lactobifido probiotics, the recent research from that Israeli university shows that they can actually compete with your endogenous bacteria if they survive through, right? So yeah, so that can actually work against diversity. And you're also putting in too much of the lactobifido DNA into the system, because in a healthy microbiome, you have less than 1% lactobacillus, right? So if you imagine, if you're taking, let's say, 50 billion lactobacilli every single day, you're putting in 50 billion lactobacilli worth of DNA into the system, and that DNA can have functionality in the microbiome. Whereas in the natural microbiome, only 1% or less of the total genome in the microbiome is made up of lactobacillus DNA. So if you're getting this daily exposure of large amounts of lactobacillus DNA, that can screw up the gene pool within the microbiome, and that may have an effect on the other organisms that are present, right?
What about spores and what about sucrose?
Yeah, so spores are different and that's part of the reason why spores, the dose is so much lower, right? If you look at megaspore, it's 4 billion. You won't find a lactobifido product that's anywhere close to 4 billion, right? They're all 40, 50, 100 billion plus. And 4 billion is the natural exposure level if you were just hanging out in the environment. That's part of the normal level of exposure that you get with the spores. Now, the spores are also different in that the spores actually increase diversity of the other microbes. Because remember, the spores can compete with opportunistic organisms, which they tend to do. And then they also produce prebiotics in the gut to increase the growth of your commensals. So we published a paper, I think it was last year or the year before, showing that when you add spores into the microbiome, it actually increases your diversity as a singular probiotic. And then, of course, the spores are designed by nature to move through. And after about 2 weeks, they move through and they go back out through defecation. Saccharomyces works very similarly as well. Saccharomyces is completely transient. It moves right through, and as it's moving through, it acidifies the gut, stimulates the immune system. It creates some competitive exclusion of pathogens. Hence, it's so useful for antibiotic-associated diarrhea, and then it moves right through. And keep in mind that the spores in Saccharomyces are organisms we're supposed to get regular, daily, natural exposure to, because Saccharomyces, if we were hunter-gatherers, we would be getting them from fruits and grasses and vegetables that we would pick and eat. And then the spores are ubiquitous in the environment, so we get them through natural exposure just by living in the land and within the land and so on, right? The lactobifido combos, the big cocktail products, are an unusual way of introducing probiotic bacteria to the system, because nowhere in nature do you get this kind of big mix of lactobifido.
This is an incredible aha. This is, and I think for all of us guys, I think this is an incredible aha moment. So thank you again. Now, the question is, for example, I think all of us should be aiming to jack up our acromantia as high as possible. So I'm thinking applying that, for example, those lactose and bifido species for so long consistently, will that harm acromantia on top of, let's say, for example, in my case, I put a whole bunch of foods that's supposed to jack up acromantia, but still acromantia is low. So meaning that, so now what you explained makes absolute sense. So in my case, for example, I will minimize at least temporarily the lactose and bifido.
Yeah, for sure. Any, especially the cocktail products, right? If there are very specific lactobifido strains that have specific functionality that you need for your, to feel good and feel normal. For example, the bifidolongum 35624 reduces the spasmodic activity in the gut, right? So and it reduces certain types of inflammatory processes, and it really helps people with IBS manage the symptoms. That one strain, usually people take them at 1 to 2 billion a day. That has perfectly reasonable functionality. It's when the probiotic industry went to these big cocktail, what I call kitchen sink products, where they throw a whole lot of species in there without a whole lot of rhyme and reason as to why each of those individual species are there. That's when you could see how these start to interfere and confuse the microbiome.
Well, what, to be honest with you, let's say the probiotic that I take has 5 billion colonies. But it's still, when, as you know, now I'm understanding consistently so basically I take it for years.
Yeah.
I think the same thing for years and years and years.
Yeah.
So this is probably the time to give it a break.
Yeah, and test it out, right? So you can change up what you do, and then in a matter of 4 or 5, 6 weeks, you can do a retest and see, hey, am I shifting things, right? And that's part of the beauty of this test. You can kind of look and see how you're doing with things.
Here comes another joke. I'm not repeating that test for the sake of the collection process. You got to come up with something that is less gross. I'm just kidding about it.
So this is the least gross of it, right? Because remember how you had to put that paper on this toilet seat, right?
Oh my goodness!
And you poop in the paper. And then you have that nice long brush, which you take the coring samples. Now, if you've done other tests, many of them you have to poop into sometimes a dish, and then you have to scoop poop out of that, putting it into another container, or you have to swab the surface of the poop. I mean, there's worse ways to do it. But
guys, I hope you all had lunches. But the thing is, trust me, as you see, it's all worth it. Absolutely, I'm repeating because I have the kids. Absolutely, thank you.
Yeah, and then same thing. This shows you other donut plots to show the distribution. And the good thing is, when you look at the family level and the phylum level, the distribution is pretty close to what it should be. So it's not that there's all of these bacteria that are off, right? What it is there's just fewer bacteria and fewer species within this microbiome, and clearly a lot of those are the keystone organisms that we saw in the summary, right? So and then when we get here to, this is again more compositional data, and really what we want to look at here is the, let's see, did we look at the pathobiome, or that the pathogen side is maybe coming up a little bit later. Oh, here it's coming
Yeah,
up.
the pathobiome, the Clostridium difficile for some reason have no idea why and how, and no effect, but Clostridium difficile is very high.
Yeah, so here's an interesting thing. So then here's the phylum level: US healthy, US healthy, US healthy. Here's the sample. The good news is all of this is within the right proportion, right? The right proportion of Firmicutes to Bacteroidetes, Proteobacter to Actinobacter, to Prevotella. What all this says is you don't struggle from weight issues, you don't struggle from metabolic issues. These are the things that typically create issues with BMI, with excess weight, with diabetes, and so on. So these are all well in line, and more often than not, we see people with this way off, especially Prevotella and Bacteroidetes. So
well, let's stop here, because all these things, to be honest with you, I am very subjected to weight gain and stuff, and adjusting even with this picture, adjusting my gut and microgut, that led, for example, in my case, to loss of over 20 pounds and feeling different. This is where the combination of the diet. This is with the diet. It's not just we're going to normalize the composition of microbiome, but the composition of the microbiome resides on what are we putting through the mouth.
Yeah, totally. So your switch in diet, switch these ratios, and when you switch the ratios, your metabolism changes, right? So that's evident here, because the metabolic proportions are pretty well in line. And again, your pathobiome index is not so bad; it's right in the middle, which means that a pathogen could be an issue within your system, but unless there's very clear symptoms, then it's not something to necessarily have to go after with antimicrobials or anything. In this case, Clostridium difficile is higher, which could mean a couple of things. It could mean I don't know when last time in your earlier in life you've had rounds of antibiotics, which again, anyone who's had multiple rounds of antibiotics throughout their life, that increases the level of C. diff. But if you don't have diarrhea, if you don't have C. diff like symptoms, then it's there, but it's a little higher than the normal index, but it doesn't necessarily mean it's causing you issues.
But
right?
guys, let's stop here for a second. For example, I haven't had antibiotics in decades, but even not having antibiotics in decades, you see what it can give, what it can do to your micro. So how vigilant and cautious we have to be, because some of them actually pop antibiotics as just it's like candies.
Yep. Exactly. And here's the thing: you'll start to see that when you start increasing your diversity, right, then this number will start going down, because the rest of the microbes will start fighting against Clostridia and bring their numbers down. Right.
Let me ask you another question, if I may. I'm so sorry,
Yeah.
I'm interrupting. It's so exciting. The effect of stress, because that's what I'm reading as of late, the effect of stress on the microbiome and the diversity.
Yeah. So one of the key results of stress is stress increases the virulence factors of opportunistic pathogens in the gut, right? Which means that when you are stressed, it actually creates an environment that favors lots of these kind of organisms. And as they start to grow, they will suppress the growth of your good commensal organisms, and then you can start to lose really significant levels of the important commensal bacteria, because you're now your biological condition is now more preferentially suited to support the growth of pathogens and opportunistic organisms, right? So continuous bouts of stress, even short bouts of stress throughout the day, can change the ecosystem enough where over time it really ends up hurting your diversity and increasing the growth of pathogens and opportunistic organisms, right? So stress is such a potent player in disrupting your microbiome. So that's something we really have to keep an eye on. So now, we want to start looking at some of the functional analysis of the microbiome. So these are parts of the test that tell you how your microbiome respond to things, right? So this is a part that's really important. So saccharolytic fermentation. Now, saccharolytic fermentation is where you've got microbes that take things like resistant starches and fibers and prebiotics from the diet, and they convert them to really important things like short chain fatty acids, for example, right? It takes lactate and converts it into short chain fatty acid metabolism.
Please tell us the importance of short chain fatty acids. Why? Because a lot of people on this Zoom, they really don't know short chain fatty acids what.
Mm-hmm. Yeah, so short chain fatty acids are things like butyrate, propionate, acetate. Number one, short chain fatty acids are the main fuel and food for your gut lining cells, especially in your large intestines, right? So in order to keep the lining of the gut integral and not permeable, you need short chain fatty acids. Short chain fatty acids also build a mucosal layer sitting on top of your gut lining cells, which is a very integral part of your immune system's function, your metabolic function, and then also your gut acting as a barrier, so your gut's not leaky. Short chain fatty acids dictate how your body responds to food. It controls sugar metabolism. It controls a hunger hormone. It controls satiety hormones. It also, short chain fatty acids reduce inflammation dramatically, not only in your gut, but systemically throughout your body as well. Your immune system counts on short chain fatty acids to not have an over response to the presence of pathogens and viruses, bacteria, and so on. There's a number of things. Your brain requires short chain fatty acids for energy. There's so many aspects that short chain fatty acids are absolutely critical for health and wellness. And saccharolytic fermentation is a process by which we make short chain fatty acids. And in this case, the production of short chain fatty acids through saccharolytic fermentation is really quite low, right? So you want to be healthy high here, and rather not healthy low. So this is where you'd want to start shifting the metabolism and really increasing the growth of saccharolytic fermenting bacteria, which are the ones that will create the short chain fatty acids. Now, so Svetlana, your diet for a while, what's your diet been like over the years? Have you gone like paleo for a while? Like what have the changes been?
My diet was vegan for the last 20 years. However, I've done micronutrient analysis and the nutrient analysis to discover that actually I am quite sensitive to few of the vegan stuff, and inadvertently I had to, let's say I'm highly sensitive to soy.
Mm-hmm.
My two highest sensitivities are soy and Brazil nuts. Hello. So I had to switch. So I'm sensitive to celery. I'm sensitive to nuts. And again, this used to be my daily thing. So I had since I've discovered in the last few months, I had to readjust my diet and introduce oily fish.
Mm-hmm.
And do different things. However, at the end of this seminar, I would love you to talk to us about FODMAPE, because I also thought, and I also think that this is an incredible thing, because what I have discovered, per se, for me, I'm a slow metabolizer through the testing and stuff, and what I've discovered that I think my cells are prone to be insulin resistant.
Mm-hmm.
So everything lines up. So again, with your explanation of the probiotics, so my food is, well, probably much better than 99% of people, because I'm crazy about the composition and the quality and the stuff. So, and I go heavy duty on my dark chocolate because there's a polyphenols for my gut.
Yeah.
Non-negotiable, and I'm not sensitive to it.
Right.
So this kind of thing. So I need to look into other aspect other than foods to actually increase things.
Yeah.
Plus, per se, living in Alberta, the food diversity during 6 to 8 months of the season is very low.
Yeah.
And now discovering that even when we get organic food, especially the food that may come from California, the water supply and everything, it still may have those traces that are very detrimental for the gut.
Right. Yep. Makes sense. So yeah, and then if you look, typically, the butyrate production, if your saccharolytic fermentation is high, butyrate production and all should be higher up here. In this case, these key butyrate producing groups of bacteria were not detected. It doesn't mean it's not there. It means that the levels are low enough where the test even couldn't pick it up. Propionate is another short chain fatty acid. Acetate is another short chain fatty acid. And acetate seems to be right here in the middle, which is good. Acetate is typically the first short chain fatty acid that's produced. So what happens is there are certain groups of microbes that will produce acetate. And then acetate is actually important because not only does acetate get absorbed, it functions in the liver, it functions in the brain, but it also serves as the starting material for the production of butyrate and then propionate. Acetate also has an antimicrobial effect in the microbiome, so it can work to control some pathogens, which is good. So it's good that the acetate production is right in the average range. But what we're not seeing is the acetate then being converted into propionate and butyrate. So those have to be increased.
And for some of us, in order to increase butyrate, for example, other than supplementing and stuff, ghee. So personally, I need to start looking into ghee. However, make sure that if you are implementing ghee, it has to be organic. It has to be full certified, whatever.
Yeah. And then this is good. I mean, the lactate production is really high. Lactate is a really important precursor to making acetate, butyrate, and propionate. So your starting material is high. So a lot of times what we'll see is that these like individuals have very low butyrate and propionate producers, but we also see very low lactate producers as well. And that becomes problematic because this is the starting material for the formation of butyrate, propionate, and so on. So you're making enough lactate, which is good, but then you also need to make sure you have enough microbes to metabolize the lactate into butyrate and propionate, because if you don't, then you can also build up lactate in the system, right? And you don't want the risk of running into lactic acidosis and other issues of overproduction of lactate. And this may be another reason why to reduce the lactic acid producing bacteria like lactobacillus. And also maybe even reduce Saccharomyces, because Saccharomyces produces a good amount of lactate, right? So you don't necessarily need any more lactate. What you need is the organisms that make butyrate and propionate to increase or convert more of this lactate. Now, the other fermentation process we talked about here, the saccharolytic fermentation. The other one is called proteolytic fermentation. Proteolytic fermentation is the fermentation of proteins, and there's beneficial aspects of fermentation of proteins, but then the other problem with fermentation of proteins is it produces a lot of toxic compounds as well, like P-cresol, phenols, ammonia, hydrogen sulfide, and so on. These things can be very damaging to the gut, very inflammatory, cause leaky gut, drive things like inflammatory bowel disease, and so on. So you're right here, average proteolytic fermentation, which is good. So you can eat good amount of protein and not make too much of these byproducts, which is fine. We see some people that may be high here, which means that they shouldn't be on a high protein diet because they're converting a lot of the protein to things like P-cresol, phenol, and so on. What you want to see is proteolytic fermentation right around here, but then you also want to see your saccharolytic fermentation right around here as well. So there's a good balance between those two types of fermentation. Amine production, which is right around here as well. This is, again, another byproduct of protein. metabolism, which is not bad. You're right here in the middle. So protein is not a real issue for you in terms of its metabolism. Phenol production, which is also good at this low level. This is P-cresol is a really inflammatory toxigenic byproduct of bacterial protein metabolism, and you don't want too much of this type of production. Same thing with ammonia, which is good. So some people will have really high ammonia producing bacteria. In those individuals, we always say, hey, you've got to start reducing your protein intake, right? Because for those individuals, protein intake is actually more toxigenic than it is beneficial. And a lot of times we see people who are on high protein diets with this kind of characteristic with having really high ammonia and phenol production. Because if you go on a really high protein diet for a long period of time, you're selecting for bacteria that make ammonia, bacteria that make phenols, and so on. So this is fine. Same thing, hydrogen sulfide. This is good. You want to be really low here because there's lots of good, healthy foods that tend to be high in sulfates, right? Foods like eggs and Brussels sprouts and mushrooms and all that, they tend to be high in sulfates. And what happens is if you have high levels of sulfate reducing bacteria, those bacteria will take the sulfates from these healthy foods and convert it to hydrogen sulfide, which actually creates massive inflammation in the large bowel. So for you, you're right down here, you're not producing any hydrogen sulfide at all. So all of those kinds of healthy foods are perfectly fine for you. So you don't have to watch out for high sulfate foods. We see people all the time that, they say, hey, I'm eating a healthy diet, but I'm not progressing with my gut health. I'm still getting lots of inflammation, so on. And then you run this test and you see that they have really high sulfate reducing bacteria. When you have high sulfate reducing bacteria, there's lots of healthy foods that seem like a healthy part of a diet, but actually to that individual, it's quite inflammatory, right? So in those cases, you have to reduce the sulfate producing bacteria, reducing bacteria before you can reintroduce some of those foods. So this kind of stuff gives you the insight on that. Methane production, again, you're pretty low, which is great. So again, consumption of proteins and all that doesn't trigger a whole lot of gas production for you, which is good. And it also doesn't produce things like TMA, which is a significant risk for cardiovascular disease, systemic inflammation, and so on. So this is good. You want to see this low as well. Now, when we get down to hormones and neurotransmitters, GABA production is not detected. Again, it doesn't mean that those organisms aren't there. It could mean that they're so low that we're not finding them in that sample. And what you really want to do is bring up the keystone species, and that will start going up naturally. Glutathione, you're great, right in the middle where you want to be. And again, these are microbes that produce glutathione for you in your gut. So you've got good antioxidant function. So this just talks through a bunch of inflammatory cascades. So a lot of the next markers are really focused on production of inflammatory compounds based on incoming dietary components. And all of these, you're low. So you've got low indole production, low estrogen, estrobolome levels. This is actually something you'd want to bring up over time, but it's not unusual to see this if you've gone into perimenopause or postmenopause, right? Because now you're not producing as much estrogen, so you're not feeding the estrobolome microbes in the gut as much, which then you start to see this level drop anyway. Naturally, if you were 30 years old and you had very low estrobolome, then we would say, okay, this is a sign of probably a hormone imbalance that starts in the microbiome. So you'd want to start to increase the estrobolome. Now, also, I want everyone to know there is an actionable step for every one of these outcomes. There is a dietary actionable step, a lifestyle, and a supplement actionable step. We just can't put that on this report to remain FDA compliant. So your practitioner, whoever orders the test, has that with them, and they can pull from that and say, okay, hey, patient A, your estrobolome is really low. Here's what the report shows you can be doing from a lifestyle, diet, and supplement perspective to improve that, right? So all of this is actionable. Everything that you see here, you can take action on. And then these are all self-explanatory functional analysis, vitamin biosynthesis, each different vitamin, B5, B6. These are all the production of methylated forms of these vitamins.
It's interesting. You see, guys, that the gut produces all these bees.
Mm-hmm.
All these bees, and those bees are essential for the cellular function.
Yep, and not only they produce it, they produce the methylated form, which is critical, right? So even if you have a methylation issue, and if you have a healthy, diverse microbiome producing all these for you,
And
it
the majority of us have methylation issues.
Mm-hmm.
Majority of us have COMT issues. I mean, not only COMT, but MTHFR issues.
Yeah. And you're unique in that you've got high levels of vitamin K2 producing bacteria, which is fantastic. We hardly ever see this. We see this tend to be really low in most people, but that's great. And that may be part of the intake of the spores, because of course the spores produce K2 in your gut for you. So that's really good to see. And then now we come to the keystone species. And again, many of these were not detected. Again, it doesn't mean that they're not there. It means that their levels are so low that we couldn't pick it up even in the test. And with each one of these, there is a dietary recommendation. One of the things that Svetlana mentioned with Akkermansia really loves polyphenols. You want to get a good variety of polyphenols. We put a couple of polyphenols in the Mega Pre that have been shown to feed Akkermansia. So you want to do that. Akkermansia also does well when you fast. So fasting can increase Akkermansia as well. So that's another way of helping Akkermansia come back to good detectable levels. Fecalum bacteria also feeds well off of certain prebiotics, similar prebiotics to what we put in the Mega Pre product. And having high levels of Fecalum bacteria is fantastic. Fecalum bacteria is the most protective organism against gut inflammation, right? Fecalum bacteria is inversely correlated with inflammatory bowel disease. So the higher fecalum you have, the more protected you are against inflammatory bowel conditions. So this is really good, high levels of fecalum bacteria, which is fantastic. Some of these rumnococci can also create skin issues. So I don't know if you've ever had a struggle with any sort of skin issues, even younger, back in the day. But some of these can be correlated. Well, low levels of rumnococcus can be associated with inflammatory conditions in the skin. So you want to bring these up. These are cellulose degraders, so they do love plant-based materials. And again, the other report provides you more specific food products that can support these organisms. And again, we have lots of butyrate and acetate producers. These have to come back up. Bifidobacterium longum is a keystone species. We can increase this significantly with the Mega Prebiotic. We can increase Butyrococcus with a Mega Prebiotic. At some point, we are coming out with a Butyrococcus product itself, and we are launching a Bifidolongum product in a couple of months that has been clinically shown to reduce cortisol and stress response, and clinically shown to reduce stress hormones and improve sleep, time to sleep, duration of sleep, and sleep quality as well. It's a
So
unique.
how is it going to be different from the other probiotics that claim to contain bifidum longum?
Yeah, so it's a completely different type of bifidolongum. It's a bifidolongum that contains an exopolysaccharide around it. So it's a unique species of bifidolongum that has this coating that it naturally produces. And in fact, in knockout studies, what we see is it's the coating, the exopolysaccharide, that does all of that function. That reducing of cortisol, the reducing of stress hormones, reducing of the inflammatory pathologies that leads to the release of glucocorticoids, and then improves sleep and sleep duration and so on. It's that exopolysaccharide. So it's a unique bifidolongum.
Hurry up. Everybody's stressed. Everybody has high.
Totally.
Hurry up.
We need it. We think we will have it launched by the end of June. And the studies on it are mind-boggling because they were all done at a research institute called the APC, which is in Cork, Ireland. It's the most world-renowned place for gut-brain studies. All of those studies have been done with some of the top researchers in the gut-brain space. And again, it's a bifidolongum. It's called 1714 strain, and it's really quite amazing. In fact, our staff has been using it. We were able to get a small shipment for the staff to try it. really is quite significant. So we're excited to bring that. But, in general, some of the most difficult things to change about the microbiome are things that you're already in good place with, right? So the proportion of the different phylum, the proportion of the different families, those are all good. A lot of the production of inflammatory things like, ammonia, methane, indoles, all of those things are within range or not at all detectable. Hydrogen sulfate, those are not even detectable. So in this case, it's really a matter of changing up diet, increasing prebiotic intake, fasting a little bit more, being outside a little bit more, being out in nature and engaging with the environment a little bit more. Those kinds of things will all start changing the landscape of these of all of these microbes.
Listen, guys, in front of all of you, I need to pin Dr. Krishnan because we're running out of time to lecture number 5. Will we? Kiran, you're going to give us lecture number 5?
Absolutely. Why
We don't have time. We don't have time to go into amazing food mate.
Yes, we do have to talk about that.
Because to me, this is like a must deal. However, before we end up, we have parents of kids. So my question is, would you recommend to test the kids? Because, this is the thing.
Yeah. So if your kid is under 3 and the kid is having specific issues, especially behavioral issues, then I would test them, because I can provide you some really strong insight as to what is off already that is driving the behavioral issues. And you can absolutely make significant impact on the kid's microbiome right at that stage, right? If they are healthy, normal kid, no behavioral issues, no severe immune issues, they don't get massive rashes and have significant allergies and all that, then there's no real reason to test at that point. At that point, I would wait till they're about 6 or 7, because that's when you start to see the adult-like microbiome really taking shape, but you could still make significant changes through dietary supplement and other manipulations. So at that point, it's a good way of looking at a preview of what they may be dealing with down the road. So if you see things that are off already at that stage, even though they may not be symptomatic of that imbalance, that's a good time to start jumping in, intervening, and setting up their microbiome in a better position. But if your kids have any issue, test them.
Some of my friends, they had kids on so many bouts of antibiotics.
Yeah. If they've had bouts of antibiotics, absolutely test them, because you need to get a sense of what was hurt by the antibiotics, right? If the kid is really high proteolytic and no saccharolytic, you want to be really deliberate about their diet. You want to make sure you're not giving them too much protein, which is going to drive more ammonia, more P. crusol, more indole production, and the kid's gut is going to get more and more inflamed and leaky. You want to start switching up the diets and so on. So there's a lot of actions you can take, and this provides you some clues as to what may be missing in your health journey, right? You're probably doing lots of things that help your system, but this provides you microbiome-related clues as to what can really, help make that difference. So that's the purpose of this test, and it really has been a game changer for lots and lots of our practitioners, and we get lots of amazing feedback from it based on that. And again, keep in mind, this is the first and only functional microbiome test, and it uses whole genome sequencing, which is the only way to detect organisms to the species level.
This was absolutely incredible, guys. We are running out of time. If you have any burning questions, please pose the questions, and so we can ask Dr. Krishnan. I am in awe. There's so much incredible information that was shared, and I hope you agree with me, and I hope you are as grateful as I am for his generosity of time, knowledge, and soul. I see people smiling. They're going to be looking at poop in a different way right now.
Exactly. Thank you. Thank you, Janet.
Here you go, Janet.
Yeah, your poop is an amazing clue to what's going on in your system, why your body reacts the way it does, why when you eat certain things, you feel a certain way, what is impacting you. And in many cases, it could be what are seemingly healthy foods in general, but because of your system, it impacts you in a different way. And getting those kind of insights can be really important, because we might beat our head against the wall going, I'm eating healthy, you know, I've cut out all of these things that are processed and bad, and I'm eating fresh foods and a good diversity, but it just so happens that your microbiome composition is converting those healthy foods into things that are not good for you. And it's good to know that, right? You need those insights. So yeah, you know, as much as you can, you know, I wouldn't test any more frequently than once every, let's say, 6 months ish. I would give yourself time to make the change. You know, if you tested once or twice a year would be ideal. And then once you've given your gut 6 months to make some significant changes, test it again to see the progress you're making. And then maybe you're good at that point. You may not need to test or make any more changes, or you might find some other insights that you could start working on. You know?